We saw how mRNA is produced from DNA. The next step is making proteins from mRNA. The subunits of a protein are called amino acids. The sequence of nucleotide bases in mRNA is translated to a sequence of amino acids in a protein. This process is called translation. When the code in a gene is transcribed to mRNA and then the mRNA is translated to proteins, we say that the gene is expressed. Not all genes are expressed in every cell. Though all your cells have the genes that code for hair color, these genes are not expressed in every cell. For example, brain cells or heart cells do not make the hair color pigments. As we saw, there are 4 kinds of nucleotides. There are 20 kinds of amino acids found in our proteins. If one nucleotide codes for one amino acid, there are only 4 possible amino acids. If a pair of 2 nucleotides codes for one amino acid, there are 16 possible combinations. 4 possibilities for the first position times 4 possibilities for the second position gives 16 possible amino acids. This is not enough either. If 3 nucleotides code for one amino acid, there are 64 combinations. This is more than enough for 20 amino acids. A set of 3 nucleotides that code for one amino acid is called a codon. Our genetic code consists of such triplets. For example, AUG codes for methionine and AAA codes for lysine. Since we have 64 possible codons but only 20 amino acids, more than one codon codes for the same amino acid. AAA and AAG both code for lysine. A gene always begins with AUG. This is the start codon where translation begins. Translation ends at stop codons. The 3 stop codons are UAA, UAG, and UGA. These codons do not code for any amino acid, so translation stops here. All organisms have the same genetic code, with very few exceptions. If AAA codes for lysine in humans, it will also code for the same amino acid in bacteria. This means that we can transfer a human gene for insulin into bacteria and the gene will still work. This is how we get bacteria to produce human insulin. A gene contains some non-coding segments which are removed before translation. These intervening segments are called introns. They are not part of the code. The coding segments are called exons. The RNA transcribed directly from DNA has both introns and exons. The cell then cuts out these introns and pastes the exons back together. The final mRNA with only coding segments is translated. If you're wondering how codons are related to introns and exons, a codon is just a set of 3 bases. Each exon or intron may be hundreds or thousands of bases long, containing many codons. You now understand how our cells use DNA to make proteins which determine our characteristics.